The endomembrane system of a eukaryotic cell refers to all of the internal membranes, and these are going to be membranes that are either directly connected together or are connected together by the transfer of what we call vesicles. Now, if we look at the structure of a vesicle is like a little bubble of membrane material. So as a bubble, it will have membrane material over the entire surface, so it's like a little sphere. And since it is a membrane, it is a phospholipid bilayer. So notice that the inside of this membrane is going to be hydrophobic because of the fatty acid tails, and the outsides will be hydrophilic. Inside the vesicle, there can be anything inside there that the cell happens to package in there. So vesicles will be like little cargo compartments where the cell can place something inside, and then we'll be able to kind of ship it around the cell wherever we need it to go. So when we talk about the endomembrane system, it will include a lot of different parts of the cell because lots of the membranes will be directly connected together or will have vesicles that can move between compartments, and the functions are going to be pretty diverse. But in general, the endomembrane system will regulate protein traffic. What we mean by regulating the protein traffic is that we will be able to ship things around the cell to wherever they need to go specifically. And then we're also going to have a lot of metabolic functions that do take place in the endomembrane system. As far as what it's actually composed of, we have talked already about the plasma membrane and we have talked about the nuclear envelope, but we'll have some new compartments such as the endoplasmic reticulum, the Golgi apparatus, lysosomes, and then also lots of other generic compartments which we'll call vacuoles that are all going to be connected together in some manner. The plasma membrane is something you would find in prokaryotic cells, but other than the plasma membrane, all of the rest of this is going to be really unique to a eukaryotic cell. So when we talk about the endomembrane system, the endomembrane means inside membranes. And in a prokaryotic cell, we don't have any inside membranes, so you would not find this system in a prokaryotic cell. So we want to start going through and talking about these parts of the endomembrane system that we haven't talked about so far. So the first one that we'll discuss is that endoplasmic reticulum. The endoplasmic reticulum oftentimes is called the ER for short. And this makes up over half of the total membranes that you find in a eukaryotic cell. So it is an extensive network of membranes. These membranes will be folded back and forth on each other to form lots of flattened compartments or sacs. And these sacs are often called cisternae. If we look in this slide, it is the blue. Notice that this one is actually continuous with the nuclear envelope. So we said that one of the characteristics of the endomembrane system would be that these membranes were either directly connected together or they could have vesicles that come off of them. And we will have two different types of endoplasmic reticulum. We'll have what we call the smooth ER, and then we also have the rough ER. Well, the smooth ER is going to be smooth in appearance, and the rough ER will be rough in appearance, but it's more than just the appearance of them that makes them different. So if we talk about the smooth ER is smooth because it will not have ribosomes stuck on the surface of it. The functions of the smooth ER will be to make a lot of lipids. So this would include a lot of those lipids that we talked about in Chapter 5, such as fatty acids and triglycerides. It's also going to be where we get a lot of carbohydrates metabolized. There is certainly a storage function to the smooth ER. Calcium ions very often will be stored inside the smooth ER, and at later times they'll be kind of released into the cytoplasm, and then we can restore them again in the smooth ER. And we also have a detoxification function. If there are poisons inside the cell, this will be one place where we can kind of neutralize those or get rid of them. The rough ER, in contrast, is rough because of the appearance of the ribosomes. So the ribosomes will be stuck to the outside of the rough ER, and these would be then some of those bound ribosomes that we described earlier. Keep in mind that the function of the ribosome is to make proteins. So essentially we have little protein factories stuck on the surface of the rough ER, which means that there certainly ought to be something to do with proteins here. Specifically, the rough ER will be involved in the synthesis or the making of secretory proteins. By secretory proteins, these will be proteins that are either secreted. These could be secreted from the cell, so they could be actually exiting the cell altogether, or these may be proteins that are going to be shipped to a particular compartment inside the cell. Additionally, we will have the synthesis of a lot of membranes take place in the rough ER. And if we keep in mind that membranes are made up of phospholipids, then the phospholipids will certainly be made here as well. Make sure you do know the difference between the smooth ER and the rough ER, and understand that the structure of both of them is to have membranes around the outside and then an internal compartment. Because they are inside membranes, these are not things that you would find in the prokaryotic cells. Now, right next to the ER, we're going to find another structure which is called the Golgi apparatus. The Golgi apparatus, like the ER, is going to be composed of a lot of membranes, and it will be flattened, so we'll have flattened sacs or flattened compartments, and it is going to specialize in further processing anything that the ER does. So the ER is making a lot of things. We said that we're going to be making phospholipids, triglycerides, metabolizing carbohydrates, making secretory proteins. But just because things are made in the ER doesn't mean that they really have the finishing touches put on them in the ER. So what will happen is the ER will be making things, and then it will package them into a little vesicle. We said vesicles are going to be like little cargo carriers. So we'll have something packaged inside there. That vesicle or compartment will be shipped to the Golgi apparatus, and the Golgi apparatus will be responsible for putting the special touches or the finishing touches onto those molecules, such that when the Golgi apparatus is done with them, they are ready to be used inside the cell, so they're ready to go. If we look at this in more detail, here we have the Golgi apparatus, and up here would be where we would find the ER located at. So keep in mind the ER is going to be a lot of those same flattened sacs, and once something is finished being made in the ER, a little vesicle will be pinched off. So if you can imagine, basically we constrict this, kind of pinch it in, then we would have this vesicle which now contains our intended cargo. We're going to ship that to the Golgi apparatus, and there will be a specific side of the Golgi apparatus where the ER products come in. This is what we typically call the cis side or the cis face of the Golgi apparatus. This would be the side that is right next to the ER. When that vesicle fuses with the Golgi apparatus, the cargo will then be dumped into the Golgi, which can then modify that cargo. That modification may be taking the protein that was made in the ER, taking the sugars that were made in the ER, and simply attaching them together. There's lots of different modifications that can take place, and by the time the cargo gets to the other side of the Golgi apparatus, it will be ready to go. So in that case, another vesicle will pinch off, containing these finished materials, and then it will ship them to wherever they need to go inside the cell. So that may be to the plasma membrane, it may be back to the nuclear envelope, it may be to the Golgi apparatus, or sorry, to the endoplasmic reticulum. But in any case, we do have a specific side of the Golgi that is responsible for shipping things out, and that will be what we call the trans side or the trans face. So the Golgi apparatus is a processing plant and a shipping plant for ER products. We talk about some other endomembrane system components inside a eukaryotic cell. The lysosome is one that you find present in animal cells. So this is going to be specific to animal cells. And this is a membranous sac, which essentially means a vesicle, of hydrolytic enzymes. Hydrolytic enzymes would be enzymes that perform hydrolysis reactions. Now, we talked about hydrolysis reactions in Chapter 5, and hydrolysis means cutting with water. So, in other words, these are digesting enzymes. Enzymes that are going to take large macromolecules, such as a polysaccharide, and digest those linkages so that we get the individual monosaccharides out of it. Or we could take a protein and we could digest the peptide bonds that make it up. So these are going to digest all different types of macromolecules depending on what's available to the cell. Now, this can be for a couple different purposes. This could be for digesting food, so intracellular digestion. As we see in this slide, we might have a membrane-bound compartment that contains some food. We would call that a food vacuole. Basically, we've isolated our food in this membrane-bound compartment. Then we would take our lysosome, which has the digestive enzymes, and we would essentially fuse the two together. As soon as they are fused together, that's going to take the food particle and expose it to these hydrolytic enzymes so that the digestion can take place. So in this manner, if we want something digested, we will expose it to those digestive enzymes. This could also be for a recycling function. So if we had some damaged components inside the cell, it would make sense to take them apart and recycle them and build something that's going to be functional again. So in the same way we did with the food particle, we would expose these to the digestive enzymes and then let the digestion take place. The lysosome and its containing of the digestive enzymes emphasizes some of the significance of having all of these internal membranes. If we did not have the internal membranes, and if we were not able to isolate our digestive enzymes, we would be unable to regulate what's actually getting digested inside the cell. So keep in mind, if the hydrolytic enzymes were just floating around in the cytoplasm, anything that they came into contact with would get digested. So this way, we carefully control that digestion. The lysosomes are found in animal cells specifically. If we look at some other generic membrane-bound compartments, these are called vacuoles. So the term vacuole and vesicle can be somewhat used interchangeably. Both of them are going to be membrane-bound compartments, and they can contain different cargo inside, and they can have different functions inside the cell. Usually vesicle we use for a compartment that's being shipped around. A vacuole would be a more stationary compartment. We can have vacuoles that contain food particles. We saw an example of that when we talked about the lysosome fusing with a food vacuole. So in that case, they're having a storage function. They're actually storing food. We can have vacuoles that store waste as well. We do have some vacuoles that are contractile vacuoles. These might be vacuoles that gain water and expand, and then they can lose water and contract. And that sometimes becomes very important for the movement of some cells. So if you have cells that change shape in order to get around, they usually do have a contractile vacuole there. When we talk about vacuoles, these can be found in any eukaryotic cell, meaning they can be present in plant cells and they can also be present in animal cells. We do, however, have one very large vacuole that is specifically found in plant cells, and this is called the central vacuole. The central vacuole takes up the majority of the inside of a plant cell, so it is very large, and its function is going to vary depending on the cell type that it's in. So whether it is in a leaf cell or a root cell or some flower cells, so where in the plant we would actually find it. It can serve a storage function. If we're talking about the roots, oftentimes food will be stored here, so maybe some starch grains would be stored inside here. We can also store waste inside this compartment. If we're talking about something like a flower cell, there may be a lot of pigments that are stored in the central vacuole. So it can serve a lot of different functions. One of the major functions that it will have is for growth of the cell. So if we do need to enlarge the size of a plant cell, this central vacuole can fill with water, which is going to help stretch the cell. And as that cell is stretched, it can then build extra material on the outside to accommodate that increase in size. So we want to make sure we understand that the central vacuole will be very large, and it will be found only in plant cells. And then animal cells can have vacuoles as well, but they're going to be relatively tiny. That's not to say that there aren't tiny vacuoles in plant cells as well, because there certainly can be. So to sum up the endomembrane system, here we see the endomembrane system. Keep in mind that these are internal membrane systems. So by an internal membrane system, we are talking about eukaryotic cells specifically. These are going to be membranes that are directly connected together, as is the case with the endoplasmic reticulum and the nuclear envelope, or they're membranes where we can take vesicles and we can move them between compartments. So we are including the nuclear envelope, we're including the endoplasmic reticulum, we can take vesicles off of that and send them to the Golgi apparatus. And then depending on what those vesicles actually contain when they come off of the Golgi, they may be called a lysosome. So if they have hydrolytic enzymes in them, we would then call it a lysosome, and that's what we see being shown in purple down here. If they were just storing some food, we could call it a food vacuole. And then we can also send them and fuse them with the plasma membrane, as we see happening down here. One thing I want to point out about this picture is notice that when we fuse a vesicle with the plasma membrane, or with any other compartment for that matter, if you were to take this membrane and kind of pull it tight, you would have added a little bit of membrane material to the plasma membrane. This is exactly how membranes are grown, or how we enlarge or decrease the size of membranes. We do that by pinching off vesicles or fusing vesicles with various compartments. Keep in mind that the rough ER is actually producing phospholipids or producing membrane material, and then we can easily send that vesicle that contains the membrane material to whatever compartment requires it. We now want to talk about two unique components of the cell that function in energy production. These components are unique really based on their believed evolutionary history. These compartments are the mitochondria and the chloroplast, and these are thought to have at one point been individual prokaryotic cells themselves. So if we keep that in mind, these are thought to have been little individual cells that at one point were probably engulfed or eaten by a larger cell. So a larger cell came along and perhaps wrapped itself around them and then completely engulfed it, so that essentially we ended up with something like this, the big cell that has what it engulfed now inside of it. If this happened, notice that when you have the engulfing take place, you end up with a couple membranes there. So we have the membrane of what was the original cell right here. And then you also have this membrane that came from the larger cell that surrounds it now in somewhat of a vacuole. And we're also going to see that we've got some various independent components inside these organelles. So if we talk about some of those unique features, one of the things that's going to be common to these is, first of all, you are going to see that they do have multiple membranes around the outside. So these are going to contain at least two membranes separating their innermost space from the cytoplasm of the cell. So at least two membranes. These will not be a part of the endomembrane system because we will not have vacuoles or vesicles that can fuse with the mitochondria or the chloroplast, and we won't have vesicles that pinch off of them either. They will contain some of their own DNA inside, so this will be one location where you do find a little bit of extra DNA inside a eukaryotic cell. And these are thought to have at once been a prokaryotic cell. because they are thought to have been prokaryotic cells, it should not be surprising that this DNA that we find inside here is going to be circular. So it's going to be similar in shape to what we found in the prokaryotic cells. There will also be some ribosomes inside the mitochondria and the chloroplast. This means that these compartments can make a lot of their own proteins, and they've got the genetic information to do that. So they are somewhat self-sufficient. We refer to this as them being semi-autonomous. So we call them semi-autonomous because they've got some of their own DNA, they've got some of their own little protein factories inside, so they do a lot of their own stuff. And at the same time, they will replicate or copy themselves independently of the rest of the cell. So if the cell requires more mitochondria, the mitochondria will produce more. So they do not have to coordinate that with the replication of the rest of the cell. So we want to know first off what's making these two organelles a little bit different from the other cellular components. And it really is based on their belief that they were once independent cells that were engulfed or swallowed up by a larger cell. We want to first talk about the mitochondria in a little more detail. The mitochondria is a compartment that you do find in most all eukaryotic cells. So there are a few exceptions to that. These are going to be quite tiny, small compartments, and you can have hundreds or thousands of these inside a single cell. So there will be quite a lot of them inside a cell. They do have two membranes. They have what's called an outer membrane, which is a normal kind of membrane around the outside. And then they also have an inner membrane, which is going to be folded. So this is the one that we see that has kind of finger-like projections towards the inside of the mitochondria. Those projections are what we call cristae. By having the projections on the inner membrane, that's going to significantly increase the surface area of the inner membrane, which will become significant in later chapters. Because we have two membranes, we do have two internal compartments, or two isolated compartments inside the mitochondria. We do have the inner membrane space, which is the area between the inner and the outer membrane. And then additionally, we have the matrix, which is the very inside compartment. The matrix is where you will find the ribosomes, and this is also where you're going to find the DNA. So the DNA is being left out in this picture, so we'll just draw in a few of those little circles of DNA. There can most definitely be more than one circle of DNA there. There will probably be several of them. As far as the function of the mitochondria, it does have a role in energy production, but specifically that will be by the process of cellular respiration. Cellular respiration will be the process that we use to break down any food molecules that we consume, so whether we're eating protein or fat or carbohydrates for that matter. And we'll talk about that process of cellular respiration in a lot of detail in Chapter 9. So in Chapter 9, we'll come back to mitochondrial structure, and we'll talk about what happens in the matrix versus the inner membrane and why it's so important that we have that increased surface area with our inner membrane. If we talk about the chloroplast, they will be involved in energy production, but this is going to be by a different method. So this will be by the method of photosynthesis. Photosynthesis we will talk about in detail as well, and that's going to be Chapter 10 for us. The chloroplasts are a type of plastid, and there are other types of plastids for sure. The chloroplasts are plastids that we find in pretty much your green plant parts. So we're talking about the leaves primarily, although there are certainly some stems that would have some of this, and maybe some fruits and vegetables that do have some chloroplast in them as well. In contrast to the mitochondria, the chloroplast is going to contain three membranes. So we will have an inner and an outer membrane around the outside of the chloroplast. And then thirdly, we'll have a membrane around these little green discs that you see inside, and this is going to be called the thylakoid membrane. Because we have three membranes, we do have three compartments. We have the intermembrane space, which is the fluid-filled area between the inner and the outer membrane. We have the stroma, which is where you will find the ribosomes and the DNA. This is going to be all of the fluid that surrounds the outside of the thylakoids. And then we do have the thylakoid space, or oftentimes called the lumen, which is going to be the interior area of the thylakoid. So that would be all the area on the inside. All right, so we want to make sure that we understand the general structure of the chloroplast, the fact that we've got three membranes, we've got three compartments, and that it is involved in photosynthesis. Additionally, chloroplasts are going to be specific to plant cells. This means that you're not going to find a chloroplast inside an animal cell. And we do want to be familiar with things that are specific to animal cells versus those things that are specific to plant cells. The cytoskeleton is going to be a network that you do find in both prokaryotic cells and eukaryotic cells. This is a network of fibers that extends throughout the cytoplasm, and so this is going to certainly make that structure of the cytoplasm more than just a liquid that's floating all over the place. So it's going to have much more defined structure to it. This cytoskeleton will be made up of a lot of different types of fibers. Some of them will be called microtubules, some will be called microfilaments and intermediate filaments. And those vary depending on their actual chemical makeup as well as their overall size and structure. And the functions of the cytoskeleton are going to be quite diverse. In general, we say that it is responsible for organizing cellular structures and activities. If we look at it in more detail, here you see a picture that shows the inside of the cell, and all of these little cables and girders that you see extending throughout that cell would fall into this cytoskeleton category. So numerous functions are here, one of which is going to be mechanical support. Mechanical support goes along with the maintenance of cell shape. So because these are extra fibers that are extending throughout the cytoplasm, it does provide some structural support to the cell. It helps to maintain the cell shape, which may be spherical in nature. It may be more box-like or more of an unusual shape. But the cytoskeleton will be involved in that. Additionally, it does provide anchorage for cellular components. So this means that if you have a mitochondria, it's not just randomly floating about in the cytoplasm. It is going to be in a specific location because it can be attached to the cytoskeleton or kind of blocked off with the cytoskeleton. The cytoskeleton is also important for cell motility. This means when entire cells need to move, generally speaking, it's going to be the cytoskeleton that's involved in that. A good example of that would be a sperm cell. So you're all probably very familiar with the fact that sperm cells have tails and they can swim. That tail will be a part of the cytoskeleton. Intracellular movement is also a role of the cytoskeleton. This would be when we have vesicles that need to move from one compartment to another. They do so along the cytoskeleton. So it's not a random floating from one place to another place and just, hoping that it hits the right location, but it will be a directed movement. So in other words, the cytoskeleton provides little trails or roads for the vesicles and other components to travel inside the cell. And that's actually what we're seeing taking place inside this little image on this diagram. One last function we want to bring up of the cytoskeleton is the regulation of biochemical activities. A lot of this has to do with the intracellular movement part. If we have components of the cell that are going to interact with each other, the cytoskeleton can bring them close together. Or if we need a lot of energy for a particular activity, then we might want to move a mitochondria over next to that. If we talk about the chloroplast, if the light's coming in on one side of the cell, then all the chloroplasts would likely be moved to that side of the cell so that they can better intercept the light that's going to be coming in. So lots of different functions of the cytoskeleton because we have lots of different fibers and things that are going to make it up. Now, if we just quickly introduce some of those individual fibers, you really don't have to memorize all the details of each one, but microtubules are one of these. These are found in all eukaryotic cells, and these are hollow tubes or hollow rods, and their name comes from the fact that they're made up of tubulin. Now we'll see quite a lot of microtubules in chapters 12 and 13 when we talk about the process of mitosis and meiosis. These are going to have a major role in moving chromosomes inside the cell when cells are getting ready to divide. So we will come back to them certainly at that point. Another type of fiber is what we call microfilaments, and the microfilaments are going to be solid rods. So these will be solid. They'll overall be a little smaller than the microtubules, and these are made up of a different protein, which is called actin. So those are two types of fibers that we have. We do have other ones as well. The intermediate ones are kind of intermediate in size between the two. And just keep in mind that they have slightly different overall functions depending on their makeup and the cell type that you find them in. If we talk about some specialized parts of the cytoskeleton, these are going to be the ones that are important for cell motility. So when we have entire cells that need to move around, they will have these special structures called cilia and flagella. These special structures are actually going to extend from the cell. And if we look at the cilia first, cilia are like little hairs that cover the surface of a cell. So when we're talking about a cell having cilia, generally there's going to be hundreds or thousands of these little cilia on the surface, and each cilia is going to basically wave around. Because they're all waving around, we don't get a real good directional movement, but we do get movement of the cell. If we talk about the flagella, flagella allow an organism or a cell to actually swim. Here we're talking about a long tail. Here we have a picture of a sperm cell. It is possible to have multiple flagella, but generally speaking, it's going to be a small number. So maybe 1, 2, 3, something like that. And again, it's a much better directional movement. So these cells can then swim towards a particular chemical signal or environmental stimulus. It's important to emphasize that the cytoskeleton is found in prokaryotic cells as well. If you look back at the prokaryotic cell example we looked at the beginning of chapter 6, you will see that that cell did have some cilia and it did have some flagella on it. So keep in mind these can be found in those prokaryotic cells, although the specific structure of them is slightly different. If we move now to the outside of the cell, and by outside of the cell, this is outside of the plasma membrane. Here we have what we call extracellular components. If we talk about the plant cell first, the plant cell is going to have what's called a cell wall, and this would be just outside the plasma membrane. This cell wall is a major distinguishing characteristic between plant and animal cells. This is something that you will find in every single plant cell, and you will never find it around the outside of an animal cell. It is composed of a polysaccharide called cellulose. Cellulose is a long linear molecule, so we get long fibers or microfibrils of cellulose that are interconnected together to make these layers of cell wall. There may be 1 or 2 layers, and it is a fairly rigid structure. So if we have a cell that has 2 layers around it, that's got a thicker kind of wall that's a lot stronger around the outside. The texture of plant materials does tell you a little bit about their cell walls. If we're talking about delicate plant materials like a petal, that probably has just 1 layer of cell wall, and it's probably a very thin layer of cell wall. If we're talking about woody tissues, so within a tree, you're going to find 2 layers of cell walls, and it's going to be very thick. This should help to tell us something about the functions of the cell wall. First of all, it is going to protect the cell because this is a rigid box that is around the outside. So it's going to protect the cell from attack from things like insects. It's also going to help maintain the cell's shape. So it's going to help protect it from mechanical forces, so from being squished, for example. Another function that we'll look at a lot in Chapter 7 is that it will prevent excessive uptake of water. Plant cells can gain water and they can stretch a little bit in regard to that, but they never take up too much water because they've got this cell wall that prevents them from doing so. If you compare that with a cell that does not have a cell wall, that cell will stretch and stretch and stretch as additional water is gained, but eventually it reaches an upper limit and it bursts, similar to a balloon would burst when you add too much air to it. So we don't have to worry about cells, plant cells in any case, bursting from taking up too much water. Now, animal cells are not going to have a cell wall. One of the reasons for this is because the cell wall is a box, a rigid box around the outside of the cell. Animal cells need to move around. They run, they walk, etc. You don't have that in the case of plants. So while plants are in one specific location and they don't get to move around, animal cells need something much more flexible around the outside. So they're going to have a structure called an extracellular matrix, or the ECM. The extracellular matrix, just like the cell wall, will be found outside the plasma membrane, and this is going to be composed of secreted glycoproteins. So by glycoproteins, we're talking about proteins that have sugars attached to them. So this is a sticky network on the outside of the cell. It does serve a lot of the same functions that the cell wall served. It's not going to be as supportive, but it does provide some support. It also is important for adhesion of cells. This can be adhesion of cells to each other, helping them to stick together. And it's also important for adhesion to a particular surface. It can be involved in movement, and it is also involved in regulation. If we have something that's going to come into the cell, the first thing it would have to pass through is the extracellular matrix. So it does play a role in regulating what gets to enter and exit the cell. Now, since we're focusing on both plants and animals, in both of those cases we're talking about multicellular organisms. And as multicellular organisms, these are organisms that are made up of a lot of different cells that are all working together in some way to run one organism. All the cells will not have the exact same function, but they need to communicate with each other to run this one organism. So there will be junctions or connections between cells that'll help them to do that. If we talk about the plant side first, plant cells are going to have these structures called plasmodesmata that run from one cell to the next. So if we draw on here two cells, these would be plant cells. Here would be my two plant cells, that's my plasma membrane that I just drew, and here would be the cell wall. Keep in mind all plant cells are going to have that cell wall. So this whole area right here would be my cell wall. This is my plasma membrane. And this right here is going to be my plasmodesmata. This plasmodesmata is a little tunnel that runs from the cytoplasm of one cell to the cytoplasm of the next cell. And because the cytoplasm is running from one cell to the next, small molecules can run from one cell to the next. So a lot of times we think about the cytoplasm of one cell as being isolated, but that is certainly not the case. With the plasmodesmata, we've got continuous plasma membrane, notice, that runs from one cell to the next. And this plasmodesmata does have to cross across both of the cell walls, so the cell wall of each of the cells involved in this. Small molecules can move through this. Large molecules are certainly not going to be transported in this fashion. If you look at this electron microscope picture, you can get an idea of how tiny the plasmodesmata are. So this would be cell number 1, up here on the top, and this would be number 2 down here on the bottom. And this area is your plasma membrane. So notice the plasma membrane is going to be quite tiny, and the white that we see here would be the cell walls. Notice how many plasmodesmata there are. There's going to be a lot of them, and having a lot of them is going to allow for better communication. So these cells will be able to communicate quite well with each other. If we take a look at the functions of the plasmodesmata, interaction and communication are significant functions, but adhering is also important because these are connections between cells, so this is going to help the cells to actually stick together. This picture right here is showing us some persimmon cells, and these persimmon cells happen to have very thick cell walls, so they make them ideal for really looking at the plasmodesmata. The cell walls are the thin white, or the exterior of the cells is the thin white that we see right here. This would represent the plasma membrane of one cell. This would be the plasma membrane of the next. And this whole area right here is going to be the cell wall of this one. Notice you've got a little dark purple line that runs from one of these cytoplasms to the next. Notice there's a lot of them. So notice that all of these dark purple lines that you see are connecting all of the neighboring cells together with each other. They're very thin, but they will allow these cells to send small molecules amongst themselves. So the more we have, the more communication we're going to have, the better these cells can all work together to run one organism. Now, animal cells certainly need to communicate as well, but they don't have the cell wall there, so they don't have to actually run a connection or a tunnel across cell wall material. So if we look at the intracellular junctions in animal cells do have what are called gap junctions. The gap junctions will be similar in function to the plasmodesmata because these will be cytoplasmic channels. The difference is they don't have to cross a cell wall. Okay, so there's no need for that. We do have desmosomes, which are going to help to hold cells together or help with that adhering function. And then we have another structure called the tight junctions, which will help to prevent the cell from having leakage or drying out. The cell wall will do a lot of that for a plant cell. So the functions of these components added together will be very similar to what we had with the plasmodesmata. In Chapter 6, make sure that you know the different parts of the cell that we discussed. Make sure you know what they're made of, if they have a membrane around the outside, if they have multiple membranes around the outside, if they contain ribosomes or if they contain DNA. Make sure you understand what the function of those compartments is. Additionally, we want to know if the compartments would be found in prokaryotic cells, in eukaryotic cells, in both types of cells, and we also want to be able to compare and contrast plant versus animal cells.